Welcome to the Echinodermata. Your host today will be this little guy, which is a feather star. This is actually a crinoid, but it has no stalk. And there are lots of these that exist now. Interestingly, though, we'll see this a little later. Most of these actually grow a stalk in their early embryonic stages, and then it atrophies and they lose it and they break free and swim around like this. Isn't that great? I think it is. There are 5 groups of echinoderms that are still extant. These are the 5. So you got your crinoids. So here's our crinoid, and this is going to be one of the stalkless versions, although the ones with stalks are still in this same group. These are the oldest of the surviving groups. You got your holothuroids, which are going to be like sea cucumbers, and there's a beautiful sea cucumber looking there. These things originally, like their ancestors, were actually they were covered in plates or had a series of interlocking plates on the outside of at least some of them. But now the plates have actually, I wouldn't say atrophied, but they've become really small. And so they have a sort of a sea of little tiny floating sclerites. They're like little miniature bones that aren't attached to anything that would make up the rest of them. Up here we have the echinoids, Echinoidea, and you can see each of these groups has a similar number of named species. They're all doing about as well as each other. You got your brittle stars, which are called Ophiuroidea, Ophiuroids. So those are all good names to learn if you didn't learn them in invertebrate zoology, or if you haven't picked them up wherever you've been, however you got here. Then the last group, the actual sea stars, are Asteroidea, and Astera means star. So that makes a lot of sense. Yeah. So there you go. Yeah. The 5 extant classes of the Echinodermata. Now, all 5 of these classes are related to each other roughly like this. So the crinoids are the earliest branch, and they have the earliest fossil record of the surviving groups. There were a bunch of things that came before crinoids that did not make it all the way to the present, or ended up branching off and turning into some of these other weirdos that you see over here. What you see is that they all start out with a similar shaped larva. It's this sort of, it's like a little pod, like a little tiny bean. And it's going to be ciliated on the outside. It's going to have a little mouth where they can eat. It's going to swim along, and it's going to undergo a remarkable metamorphosis. It'll go through a stage where it has these little spines sticking out, and some of these are then going to become spines actually on the outside of the organism. And it will then mature through a dazzling series of metamorphoses into the adult forms, which you can see here. But you can tell from the larvae that they're all actually fairly close to each other. And since these are close to humans, we are both deuterostomes. That makes us a lot closer than either echinoderms or humans or any chordate would be to something like arthropods, like insects. Yeah, we're pretty far from them. There's actually some useful genetics that can be done on these. Now, people find ways to do useful genetics all the way down to nematodes. There are uses for other things, but these actually are pretty widely used in developmental biology, because you can study the processes and they happen really fast, whereas it takes much longer for chordates like us to develop from babies to maturity, like a mature tiny adult. Inside of echinoderms is one of their defining features. It's going to be a water vascular system. So they'll have a separate container for all their actual body parts, organs and things like that, and a separate set of, instead of blood, it's a water vascular system. Now, it's not just seawater. The salinity is actually a little different. And it does have its own little thing, kind of like an anus, madreporite. So waste is going to be squeezed into this by various cells and various tissues. And so it's a way to get that out, but also it's basically hydraulic. So the organism can squeeze some of these larger chambers, and it can actually push the water into some of the smaller chambers. And so the tube feet that you would find on something like a starfish are inflatable. They can be inflated and then retracted, and that helps them to grab onto things. And this is how these animals are going to move. Also, notice the arrangement. So the tube feet, which are right down here, are going to be paired. There's going to be two of them beside each other running down a little track. It looks like a little runway, just like you'd land your plane on. And they're each going to have these little sort of bulbs at the end that can be squeezed with muscles to inflate the tube foot, squeeze the water out of the bulb and into the tip. Yeah, and by a coordinated action of these, they can actually move themselves along. These also serve as the food delivery device. So these tube feet are going to grab onto food, and they will then funnel it, again through coordinated activation, basically passing the food from one to the next, all the way down to the mouth. And then the mouth is going to be here. The mouth is in here. Some of these starfish are actually going to extrude a little pouch to start to digest things, because why not do some of your digestion outside of the body? Yeah. All right, so that's enough just basic echinoderm stuff, just as a refresher, and just so when we're pointing to the parts on these things, you can say, oh yeah, I know where that is. I know what that is. This is called an ambulacra, like ambling, like lets you walk along. But in this case, it's also the thing that moves food along. There are ambulacral grooves running down each of the arms of a sea star. There are ambulacral grooves running down each of the arms of a swimming crinoid. There are ambulacral grooves that basically form the mouth parts of sea cucumbers. And there are ambulacral grooves that run down the sides of echinoids. So they have a sort of a biting mouth at the bottom, but they have these ancestral ambulacral grooves that can catch food and deliver it along on the sides. Yeah. So yeah, it's there because it started out there. Here are some of the earliest echinoderms. These are in the crown groups for their classes. So this is a stem group echinoid, and so echinoids are like a sea urchin. They're going to be a little ball made of little plates on the outside. Can you see the ambulacral groove? Of course you can. It's right there. Do you know how many of those you're going to find? On modern echinoderms, 5. Always 5. On ancient ones, 3, 2, one shaped like a Y, all kinds of weird stuff. Don't worry about those yet. We'll get to them. Here is a little stem group crinoid. So this one has no stalk, and the cup is really tall, and it's got little tiny arms. It's just figuring out how to do arms. Isn't that adorable? Here is the mouth apparatus of a holothurian relative, so a sea cucumber relative. And these are some of the plates that actually were covering the rest of this creature, which do not exist in the modern ones. Here's an early thing on the way to becoming an ophiuroid or a brittle star. And here is a fossil true starfish asteroid that you can see down here. Although it has some differences, because all these things are early, and we're working all of this out. The most iconic echinoderm, the starfish, is actually the last of these groups to appear in the fossil record, we think. We think they are very recent. And crinoids are the oldest of the old ones. Actually, echinoids are very recent as well, depending on where you draw the line for, when it's echinoid enough. They're doing things that you find over here, but they're doing them in a different way. Yeah. Okay. So now let's look a little bit at development. So during development, these are going to be deuterostomes, which means they're going to make the anus first and the mouth second as they're going through gastrulation. That's where you take a little hollow embryo and you basically punch a tunnel up inside of it so that it has a hollow tube in the center. So it looks like that, but with a hollow tube. Yeah, my fingers don't work quite well enough to do that. And so the first bit that's punched through is going to be the anus, and the second one will be the mouth, become the mouth. So these will have a little larval skeleton, and it's a mesodermal skeleton. It's going to be made of stereom, S-T-E-R-E-O-M, when it is fully mineralized. And here you can see the arrangement in 4 of the groups. Crinoids were studied less and kind of later, but there are a couple of groups that do study these, which makes me very happy. Yeah. There's one kind of crinoid that's relatively easy to access over in Japan. I know there's a Japanese group that works a lot on these. I'm glad there is. So we know something about echinoid genetics because fairly early on in the game, Strongylocentrotus purpuratus, which is a big purple sea urchin, so the ball with the spines on the outside, that was sequenced. And they found inside of this all kinds of transcription factors, which turn on other genes. And so they mapped out how these genes get turned on. And when one gets turned on, this will go down and it'll repress this other one, stuff like that. So they've got this nice little network. And this is a thing that you're going to find with some modification throughout the Echinodermata. Leading to some of these downstream genes that will eventually be turned on, and those will start to actually remodel the organism in some cases. But yeah, have a look. There are a lot of gene families that you find in echinoderms that are actually associated with the immune system in us. Doesn't necessarily mean that's their function, but I think in this case, at least for a number of these, that is their function. And so what are these things? Let's unpack them. So C1q, C2, C3, 4, 5, those are the complement system. This is the stuff that's floating around in your blood. And we share a common ancestor with sea urchins. And so we're all working from more or less the same equipment. So Homo sapiens, in a way, has fewer of most of these components than the sea urchin. So we have a little bit of a simpler, not to say useless, but a little simpler complement system. And there are parts that we are missing that you find in, for example, Drosophila, that's DM, the fly, and Strongylocentrotus, which is the echinoderm. Over here, HS is, that's us, Homo sapiens. CI is Sionia intestinalis. It's a little primitive chordate, but sort of squishy. Yeah, like a Staricidian relative. Yeah, one of those things. Oh, and CE is Cenorhabditis elegans, little wormy guy. All right, so yeah. But then look over here. Like, okay, there's a lot of those things. I think are scavenger receptors, but look for these TLRs. Do you remember those from immunology class? Those are toll-like receptors. Look how many we have. 10 plus 1, the little psi means a pseudogene, because ours got broken and we can't fix it. Yeah, evolution breaks things. Yeah, there's usually no fixing it. You just make a large change and hope it works out, and usually it doesn't, but occasionally it does. Compare that to 222 of these in Strongylocentrotus. That's really something. Toll-like receptors, just for your refresher, are these little proteins. They're going to be located. Some of them poke into the internal chambers of a cell. So they're going to be studying whatever the cell has just ingested. And some of these are going to be located on the surface of the cell. So they're going to be monitoring the environment around the cell. These things are going to be keyed to bind to different types of essentially danger molecules. Like if there's loose DNA floating around outside of a cell, that means a cell has exploded somewhere. And the closer you are to that, the closer you are to whatever made that cell explode. So this is a sign of bad things. Yeah. This is partly why, or goes hand in hand with apoptosis, where the cell will shred its own DNA if it is in the process of destroying itself, so that it doesn't set off the toll-like receptors of all the cells around it. Because, that's just common courtesy, isn't it? I thought it was, yeah. Some of these things will bind to stuff like lipopolysaccharide or the flagella of a variety of bacteria. and various other things. And so, yeah, here they are. And then the result of these things binding to their target is that a message will be sent, which basically is a kind of an alert for an immune cell without getting at all technical about it. But it will set off a program of actions, and it will make it possible for cells to go into a kind of a defensive mode. And it will generally result 50 steps down the line in making it harder for that cell to be infected and harder for cells around it to become infected by some sort of infectious agent, be that bacteria, viral, whatever. Yeah. So there we go. All right. What else have we got in this crazy, crazy sea urchin genome? Because sea urchins, they're people just like us, I guess. Yeah. A bunch of genes that are used for apoptosis, definitely conserved, and you'll actually find far more copies of most of these in Strongylus entrotus than you find in Homo sapiens. So they are lapping us when it comes to apoptosis, which you would need to do a bunch of apoptosis in order to change the shape of the body that many times during all the metamorphoses. They are growing and getting rid of body parts in a very controlled, programmed way. And I don't know that you necessarily need more genes to do that, but you need a lot of different ways to control them. And so it may be that this abundance just reflects a variety of very specific needs for control, different types of control. You know, like they're all hammers, but they're hammers that are sort of key coded, so you can only pick them up at a certain time. Yeah, that's speculative, but yeah, that's how I see them anyway. But yeah, a lot of genes that you would find that look right at home inside of our histone metabolism or RNA metabolism or cell cycle control, little cyclins and cyclin-dependent kinases. So that's cool. All right, now let's go in our way, way back machine to like the Cambrian, I guess. Yeah, why not? And we have some of these fossils that you can look at. So here are some of the super early echinoids. And remember how I said that echinoids, they look like they have five-fold symmetry, but actually it's two-fold symmetry. Actually, in the stem group, it's asymmetrical. There's zero-fold symmetry. They're just out there doing their thing. So on this one, it's sort of wrapped around. It kind of looks like a little beehive. And this is the actual ambulacral groove right here. It sort of splits right here, if I remember right. And so it ends up being Y-shaped and wrapping around the body of the organism, because why not? Yeah, why not? This is like an early relative of things like crinoids. These guys are, you call them carpoids or cystoid. They didn't make it, but these used to be the dominant kind of echinoderms. These were the ones that really got everything going. We think they lived along the bottom, and they would have an appendage for kind of pulling themselves along or pushing themselves along. And they would also have sometimes appendages up at this end for sort of scooping food into the mouth hole. Because, yeah, that's what you do. So here's a little pattern for the way that these organisms have developed over time, the way they evolved. Here we're looking at Hox genes. These are homeobox genes. They're involved in limb patterning in particular. And so here we have organisms that are definitely not bilaterally symmetric, but they are related to organisms. So an earlier branch would be the hemichordates. And there's a particular kind of worm that's a hemichordate. And so they have the similar Hox genes. And so you'll see these things going in a particular direction. And it's actually a set that you find preserved almost exactly in crinoids. So between hemichordates and crinoids, there really isn't much difference in terms of the organization of their Hox genes, even though the Hox genes are making a five-fold symmetric crinoid with ambulacra versus making a hemichordate worm that mostly lives burrowed in the sediment. Yeah, very strange. But it's the same patterning genes. It's just they're having a different outcome. So this is part of the puzzle, definitely not all of the puzzle. And then what you'll find in the modern echinoderms will be that there will be extra copies of Hawks 11/13. And so those will show up over here. Those are the ones that are in gray. So we don't know what the Hawks gene clusters looked like. And it is a cluster of these little genes that get turned on in a specific order and at specific parts of the body. They have responsibilities, if you like. Or rather, the zone where they're turned on will eventually become the head or a limb bud or something like that, internal organs. Without knowing this, you can still say that, wow, since we have this organization conserved all the way through, there's asteroids, they're only missing Hawks 6. We don't have a good, or didn't have a good genome at the time of this publication for brittle stars. That's why they are broken apart. We had a transcriptome, so we could tell that these genes exist, and so they're putting them there in that order, but that's why there are dotted lines between them. But yeah, basically the same thing. You only get this rearrangement when you get to the Holothuria and the Echinoidea, and then they are both going to have, or rather the Holothuria won't have it. They have the normal one. Echinoids have this crazy orientation because they're one of these very late branching groups where they're just going to move this chunk down a little bit and flip it over. And so you see that this whole area is basically flipped like that. So there's 5, there's 11/13, there's 5, there's 11/13. So just flip that whole chunk and you've got it. Just an inversion of a segment of the genome. It even ended up in the same spot. It just got inverted. And that's going to be common to the echinoids that have been sequenced at the time. Yeah, and there's still a lot more echinoid sequencing to do. Believe you me. Yeah. Here is that beautiful Japanese crinoid, Metacrinus rotundus. Yeah, looking fantastic. Like on starfish, most of these organisms, at least the extant ones, can regrow limbs, bones and all, if they are lost. As long as at least part of the brain, which is shaped like a ring, is preserved. But it doesn't even have to be all of that. And so who knows what that means for like starfish cognition if you lose nearly half your brain and regrow it. Yeah. Were there memories there? Do starfish have memories? Do they have personalities? Is half your personality gone? Do you get a new personality? I don't know. Probably none of those things. Probably none of those make sense. But you can see this one had a couple of its arms sheared off over here, and they're starting to regrow. So good for you, crinoid. Regrowth is a positive thing. Here they're showing a dissection where they're actually going to test out the regenerative properties of these crinoids. So they're taking the living animal, they're making cuts down there in the stalk, and then they will cut it at different points and see if it can regrow. And then as it regrows, so they'll cut it right here below the calyx, will it start sprouting pinnules? And then this is basically what happens. It sprouts pinnules there, and then it starts to make smaller ones that are closer to the top, and it's going to keep putting in segments. And so you can follow this segment as it moves down. The region of growth is going to be right here next to the cup, which is kind of an interesting way of doing things. So the distal part is actually going to be the oldest part on this regrowing crinoid. And so they're hypothesizing that this is the way that it normally grows. Anyway, you're just seeing it in action because it's part of the repair process. All right. Crinoids like to feed on little particles. And so this is another experiment where they took these crinoids, they're growing them in a lab, and they're actually feeding them different kinds of food. So some of them will be actual crustaceans. They'll feed them that for nutrients, but they'll feed them different sized beads, and they'll see how many of these beads end up stuck to the crinoid. Because the crinoid can't afford to be like thinking about what it eats. It catches what it can, and if it's the right size, it shoves it in its mouth. And as you can see, it kind of avoided the small little little balls. Only got a couple of the medium size. Many more of the crustaceans. So even though they are bigger, it was able to probably specifically taste them, and probably with the tube feet or some adjacent sensory organ. So it's able to select what it wants to eat. And it did not eat all the little beads. But then it had a real affinity for the smallest ones that were here. And so these were phytoplankton. So what they saw was that the crinoid was careful about what it eats, just like, I don't know, maybe your kids someday. It really likes these little plankton, basically, zooplankton. Really likes these little phytoplankton, little photosynthetic things, and it more or less rejected the two kinds of little styrofoam beads that they tried to feed it, which, good for you, crinoid. Yeah, way to discriminate in what you eat. So it's not just catching anything that comes by, it's catching, selecting, and probably selectively releasing some things that it didn't want to catch. Okay, now let's talk development. So here is the early development of a crinoid. And here's our little larva. Here's a much smaller stage, so it's actually breaking up into individual cells. It's going through gastrulation right about here. And then this little larva has the cilia patterned on the outside and some little color patterns developing. Yeah, this is going to go on through those metamorphoses to form the rest of the organism. So let's look at these ciliary bands. So we're looking at this at different stages of development. Doliolaria is the name they give it when it looks like this. And so they have names for each metamorphic state. Diplurula and semi-doliolaria, and then proper doliolaria stage. And yeah, you can see the little ciliary bands that run around. And there's a little mouth, and the ciliary bands are going to point in toward the mouth so that it can feed itself as it swims along. But yeah, it's just like it's got a little fat belly, basically, and it's growing in there. It's developing. It's rotund. Yeah, it's a nice way of saying it. Here is a poromitra. There we go. So this is one of those non-stalked / swimming crinoids, and we're looking at the adult here. So let's orient ourselves. These things down here are cirri. These are going to be at the bottom of the picture here. They can be used for grasping. They are under muscular control. And you can see that they're in groups of what appear to be 5. So there's 5 at that level, 5 at the level above, 5 major arms. Yeah. Sometimes the arms will branch, and so it'll look like 10, but that's the basic idea. Here is the, so that would be the ab-oral side, where we're looking at the back of it. Here's the oral side. There's the mouth in the center. The ambulacra are going to run down these, so each arm is essentially split into 2 arms, but each one just connects up to one of 5 ambulacral grooves, and then those go toward the mouth, and there's the mouth right there. Yeah. So let's look at some of the development of a poromitra. And here you can see it actually starts out, it's got a little egg, basically. Here's the sperm and the egg coming together. And here's our little larva. It's starting to gastrulate. It's starting to develop. And yeah, the ventral surface is V, the anterior is A. And you can see these things. This one's sitting on one of the arms of a larger crinoid. And look inside this little case there. And there you will see, look, it's got a little holdfast on the bottom. It's got a little cup. It's got all the right plates. The infrabasals are tucked away underneath. Those are the basals. These are the radials, just like in crinoids, because it is a crinoid. And it has a stalk. And as this thing grows, it is going to lose that stalk. It is going to cut off that stalk and not look back. But first, it's going to settle itself down. So here we are, settling down, suctioning on, basically with our adhesion disc down here at the bottom. You can start to see the little feeding arms here. And they've got lovely close-ups of the calyx with the basal plate and the radial plates. And then we've got some of these basically arms that would be sitting on top of it. Yeah, and there you can see the early splitting in the actual arms. And so that's where they're joined together, but they split very early, and then they're each going to become essentially two arms out of the one. Those will then, lose their tail, and basically the head and a couple of cirri from right down here are all that is going to be carried on through the rest of life. Here you can see some of these things. This one is non-reproductive. This one is reproducing. And so it's got, look at these sort of swollen little pouches here. And this one's using its cirri to hold on. That's what the C is. And the swollen little pouches are going to contain the new little eggs. They're the ovaries. And they're going to release the new eggs and start this whole little process over again. So some of these pinnules have reproductive activities, some do not. But the pinnules on the other side, that's where the ambulacra are going to be. And so those are still going to be feeding right down the center, but we're going to be growing babies off to the side. Yeah, it's a good way to do it. And so, yeah, these this particular kind of crinoid, they are either male or female. They're not hermaphroditic. And they both have these little swollen glands, which will then either release sperm or eggs. So they can then, fertilize and have more little baby crinoids. Who doesn't want more little baby crinoids in the world? They're great. Oh my gosh. They're fantastic. So there you go. It's a good place to leave you with the little stemless crinoid and its little baby stem. And it has it there because in its development, in the evolution of this group, the early ones are all stalked and they're rooted onto the bottom. And that is an ancient trait. And so the only way to get to a stalkless version, like you see now, is to go through all the steps and then cut off the stalk. It's why baby humans, when we're little embryos, still have fish tails and fish gills. There is just not a good way to get rid of that at the time because it serves a real purpose during development. And you can always do apoptosis on it or resorb it later and make it go away. Yeah, why not? So there you go. That's been crinoids. I hope you like them as much as I do, but that would be hard because I like them a lot. All right.